This is Group 2, Expression and Biochemical Characterization of a Type 1 Methionine Aminopeptidase of Plasmodium vivax. Today, we will be discussing the topic of protein expression, purification, and biochemical characterization of a Type 1 aminopeptidase of Plasmodium vivax. The overview of this topic is presented by Group 2 of Texas A&M Commerce of Dr. Nair's Biochemical Characterization Method 1 class. Group members include Nikki Burt, Jill Clay, Terry Essie, Stacy McDonald, and Nicole Morgan. In this discussion, we will define a MET-AP, what its critical role in protein is, how deletion or inhibition affects the protein, and how it can be used as a target in developing drugs. Methionine aminopeptidase, or as we will refer to them as MET-APs, are enzymes that play a role in nascent protein maturation. Removal of these N-terminal methionine residue from newly synthesized proteins is an essential post-translation process. This removal is required for proper functional regulation, subcellular targeting, folding, and protein activity. Type 1 MET-APs play a critical role in the maintenance of cellular physiology and organism survival. How exactly do the MET-APs affect the organism? Depending on the MET-AP's deletion or inhibition in bacteria and yeast, the organism is either inhibited in growth or a fatal process commences. The type 1 MET-APs we are looking at are a new target focus for developing drugs against pathogenic microorganisms like malaria, which we will be focusing on in this presentation. Specifically, we will look at MET-APs in the malaria parasite Plasmodium vivax. Malaria is a devastating parasitic disease transmitted through the bite of infected mosquitoes. Plasmodium vivax is the most common of the human malaria species and is becoming increasingly resistant to malaria drugs. Pictured is the parasite P. vivax and its life cycle, which occurs in the liver cell. Plasmodium vivax is of research interest due to its highly infectious nature, causing 70 to 80 million cases annually, making it a major public health concern due to its ability to relapse. This parasite is difficult to control and reemerges from areas where it has previously been removed. The goal of research is to use methionine aminopeptidase, or MET-APs, to block the metabolic pathways of malaria parasite Plasmodium vivax. To express and purify the enzyme PBMET-AP, it was amplified by polymerase chain reaction. The product was product purification occurred over several steps of ligation, digestion, centrifuging, and transformation. The final product was confirmed through SDS page, proving that the PBMET-AP1 protein was produced and isolated. The tests for optimal activity at different pH values, PBMET-AP was added to several buffer solutions, and the activity was determined by measuring the hydrolysis of a fluorogenic peptide substrate, which released fluorescence with a specific excitation and emission wavelength. After testing activity in buffers ranging from a pH of 5 to 9, pH 7.5 revealed optimal enzyme activity. Further tests for enzyme stability showed that acidic conditions and temperatures above 50 degrees made the enzyme unstable. Stability was greatly in temperatural conditions between 4 to 37 degrees Celsius. Enzyme activity was also tested in different concentrations of various metal chlorides for 20 minutes at room temperature. After seeing a high relative activity in manganese 2 chloride, further testing was done at various concentrations showing optimal activity at around 1 millimolar concentration. At concentrations above 1 millimolar, activity decreases, which suggests that binding of excess metal ions to the active site region results in inhibitory effects. Effects of inhibitors and substrates specificity was tested through several assays. The PV-MET AP1 was pre-incubated with each inhibitor, then the substrate was added and enzymatic activity was measured using the same fluorescence assay as before. The only effective inhibitor tested was EDTA, as seen in the middle of our table. Since the enzymatic activity of PVMET AP1 is inhibited with EDTA, further tests showed how activity can be restored by reintroducing and incubating the MET-AP sample in 0.1 millimolar or 1 millimolar solution of manganese 2 chloride. Substrate specificity was analyzed against several fluorogenic peptide substrates. PV-MET-AP1 showed high specificity for N-terminal methyamine. This information would be helpful in future studies in the development of anti-malarial drugs. In conclusion, being able to successfully express and characterize PV-MET-AP1 allows researchers to better understand the fundamental biochemical nature of the enzyme. Although physiological and functional significance of PV-MET-AP1 are not clarified in vivo, knowing how the enzyme acts in nascent proteins and its similarities to other MET-APs will aid in future research regarding this topic. These are the sources we used. Thank you.